Hello and welcome to Psych319, Chapter 2, Part 3. I've been talking about various brain systems. Now we're going to talk about the hypothalamus. So here we have a picture of the hypothalamus located in the midbrain. I'm going to zoom in on that picture, and you can see that the hypothalamus is actually a group of nuclei, hypothalamic nuclei, that largely serve a common function. It controls the autonomic nervous system and the endocrine system, which are involved in the 4 Fs, as we joke, the fighting, feeding, fleeting, and mating. All the other organisms in the picture are just really for orientation. The optic chiasm is also irrelevant. That is where the nerves from the eye cross before projecting at the back of the brain. So all the colored structures are what I'm referring to there. Um, the pituitary gland is also related to this system. Zooming in on that, the pituitary gland sits beneath the hypothalamus, and it is a structure that is highly vascularized. There's a lot of veins and arteries running through the structure. And so what happens is the hypothalamus secretes some type of command for your body to, let's say, flee. You're being chased by a wild animal, and so you need to become very alert and active rather quickly. So it secretes there are neurosecretory cells that exist in the hypothalamus, and they cause certain hormones to be secreted in this vasculature, and that gets circulated throughout your body via the pituitary gland. So blood vessels connect to the anterior pituitary. Blood vessels connect to the anterior pituitary with the hypothalamus, and these cells secrete hormones that again stimulate the anterior pituitary to stimulate and create its own hormones. And those go around the body. So the hypothalamus produces hormones that are carried to the posterior pituitary via another process, but you don't need to know that. So what happens when the pituitary goes wrong? Well, over or underproduction of the growth hormone results in growth problems, and gigantism is the most notable of these because it's kind of hard to miss. This used to be, although he's not anymore, used to be the tallest man in the world from the Guinness Book of World Records, and standing next to some rather normal-sized females in a car as a result of a pituitary growth disorder. So let's move on to another structure, the hindbrain. So here we have the back view of the brain and the cerebellum, and you can see a cross-section. This would be a coronal section of the hindbrain, and there are several structures involved here. Cerebellum, as we mentioned, was involved in learning and motor control. And then the pons are the other structure I want to mention that are responsible for sleep and arousal. The medulla oblongata is very important. It's responsible for respiration and cardiac control. So you can sort of think of this as the on-off switch. If this is on, you're alive. Congratulations. If the pons are turned on, then you're awake. You're alive and awake. And then the further you ascend throughout the brain pathway, then you introduce things like other switches that you might be awake, but you might not be attentive. So if a certain switch, perhaps in the thalamus, which has been involved in attention as well, if that is on, then you are not only alive and awake, but you are attentive. So I'm going to do a very quick and broad survey of a bunch of research methods that are used in neuroanatomy and psychology and cognitive psychology and cognitive neuroscience. And again, you don't have to know all this stuff, but I just want to let you know what's out there. So we've learned the most probably in psychology from experimental ablation, and whether this was something that humans inflicted on a brain to learn about it, or it was information learned in the battlefield after somebody suffered from a gunshot wound. It's all the same. You know, some part of the brain gets injured, and we learn about how that injury affects human behavior. So we say a lesion is an injury to the brain. Lesion studies inflict lesions to determine where what the injured region is responsible for. There, again, remember, there's no specific brain region is solely responsible for a behavior. Usually it's multiple regions that coordinate together. So brain regions tend to have functions that contribute to a behavior. And then sometimes you can do something called a sham lesion, which is a control condition where a surgery is done without actually damaging the brain. And that way you can determine whether the behavior was a result of the actual damage to the brain or just the mere effect of undergoing the surgery. So here is a lesion. The lesion is not actually these large gaping holes. These are the ventricles, the third ventricles, which I referred to earlier in this talk. So that's where cerebral spinal fluid would be. The lesion is actually here, and you can tell by not only the bright glowing red arrows, but the fact that the cell density dramatically drops off when you get to this in that area. Sometimes you can create a lesion by using aspiration, which is sucking it out with a vacuum, literally sucking out brain tissue. You can put a little electric wire down there, and that wire you can pass current through it and kill cells that way. You can use radio frequency current, passing radio waves of particular frequency to do that. You can also use different types of acids, excitotoxic lesions. So these acids typically cause neurons to fire so much that they die. So they become hyperexcited, excitotoxic, and then not only do they die, but they start to kill off some of the cells that are immediately connected to them. Recently, we've developed a great technique called reversible lesions, and it's you can take an electrode, a very, very small electrode, and not only introduce a recording electrode that can record the activity of neurons in the brain, but you can also introduce a cooling probe, and that cooling probe will, oops, let me go back a second. That cooling probe will essentially bring the brain to a particular temperature where the neurons stop working. So you freeze the neurons, so to speak. They stop working. But the cool thing is that when the temperature goes back to normal, the neurons start working again. So you bring it to a temperature where they stop working temporarily, but they can come back. And so you can see behaviors come and go with the introduction of reversible lesions. And this is a demonstration where cooling probes were used. Here we have a cat, and the cat, I believe, has a, see if I get this right, they have a really high incentive reward stimulus, like something really tasty like canned tuna on one side, and then they have something lame right in front of them like kibble. And you can use a cooling lesion to essentially make the cat blind to a particular part of the visual field. So here's the cat exposed here, and normally the cat would beeline itself right to the high reward stimulus. It'd say, "Ooh, I see tuna, delicious." But in this case, when you turn on the cooling probe, you can actually get them to ignore that high reward stimulus. They're actually blind to the right half of the visual world. So they can't really see anything out here. And they pass up that high reward stimulus and go for the low reward stimulus, the kibble. But the second you let the cooling probe back, or you put it in the left side of the visual field, they might be blind to everything over here, but they all see that high reward stimulus and they'll go straight for it every time. So really, really cool technique. So a lot of the things that I've been alluding to the fact that we do lesions and record from electrodes, and a lot of the stuff happens when you're doing stereotaxic surgery. So here is a rat in a stereotaxic apparatus, and they have levers and knobs that allow you to position the electrode anywhere in 3D so that you can precisely target a particular brain area and do that again and again repeatedly. So it's a very, very accurate technique. The rat is held in there with a bite bar and ear bars. The rat is anesthetized during all this. It's not like they're doing this while they're awake. So the rat doesn't know what's going on. It just wakes up with some sutures in its head the next day. So you can do this to induce a lesion. So all this stuff is in your notes. We can also do this in humans. We do this anytime a human is going to undergo brain surgery. We want to make sure we're getting the correct area of the brain. You don't want to lesion the wrong area. So this is the same type of stereotaxic apparatus, a little more elaborate, but same idea. Instead of ear bars, we use bone screws, a halo that sort of holds the head in place. And the difference between rats and humans is most often the human is awake during this procedure. They have some local anesthetic on the skin, but they can't feel anything because there are no pain detectors in the brain itself. The brain is command central, but it doesn't have any ability to detect pain inside itself. So you can go in there with a guide tube and lesion or record from specific parts of the brain. To make this a little more accurate, we usually perform some type of neuroimaging. This is a multi-layered MRI image that combines different types of MRI to allow us to look at the major blood vessels so we don't accidentally pop one of those using angiography. You can get the black and white image here is the anatomical MRI. The CSF is represented here in blue. Major fibers of passage, white matter bundles are in yellow. I believe it's neuronal activity is over here. I think I got that. Yeah, no, this is the tumor that they're trying to remove here. And then you have some neuronal activity that we might map just in case we are going through an area that's really important for language or something. We don't want to destroy any of that brain material. So the surgeon might go through the side here so that they can, maybe underneath so that they can get to the tumor without going through this major vasculature or through some of these important brain function areas. There's also an anatomy. So we refer to when you're doing anatomy in tissue slices, we call that histology, and this is a microtome. So what you would do is you would take a rat and you would record from its brain or lesion its brain, kill the rat, freeze the brain in a fixative solution, and then slice the brain in using this blade here with very, very thin slices, and then expose that to a chemical that will highlight some particular protein or cell body or something like that. So we used to use that for a really, really long time and still use it to get anatomical information. This is stuff I've already said. And you get beautiful images like this. So this is a cell body histology, a very old technique developed by Golgi. This is how we first saw cells. So again, a very thin 30 to 50, maybe at most 100 microns thick. And you use a solution called methyl blue or cressyl violet that stains a particular part of the cell body called Nissl substance. Now zooming in on that, so here we have a magnified image of that, and we can see individual cells. It's really, really cool. This is probably a pyramidal cell, judging by its shape. Other histological methods include electron photomicrographs. Electron microscopy is what it's called. So instead of staining the cell, you can slice a very thin tissue, bombard it with electrons, and then you'll have a plate on the other side of that slice that receives the image. As the electrons pass through the slice, they hit the plate, and all the data is recorded, and you can see very small structures within the cell. So here we have a synapse. You can see these tiny little vesicles. Those contain a neurotransmitter. Here we have mitochondria in the cell right there. If you look closely, you might even be able to see things like chromosomes. Really cool stuff. All right, that's my duck timer. So I'm going to stop there and continue in the next slide. And until then.